formulate the differential equation in two dimensions, in x and y directions. Here we see rate of change in the calcium concentration. It is depend on the diffusion of the calcium concentration, and R is a reaction term which due to the calcium buffering. Another is JVGCC. It is calcium entered into the cytosol and increase the calcium concentration. That's why it is taken plus. Sorry, RJ should be minus because it's decrease the calcium concentration. And P out is exertion of the calcium concentration outside the cell. This follows the law of mass action. Here we have used the fecal diffusion law for the diffusion process. This is the formulation for the voltage-gated channels. We use Goldman-Huskin models, and we know the Goldman-Huskin won the Nobel Prize. They are mathematicians in the neuroscience. Currents is changed into the flow of the ions. Using that equation 7. Volume of the astrocytes, because here we use the astrocytes. Maybe for the analysis depend on the volumes. After putting all these values, we find equation 8 in the steady state conditions. Here I am taking only the steady state conditions. These body conditions I have used for x is equals to 0, y is equals to 0, that is the flux conditions, and all the rest areas x is equals to 5 maximum, it is 0.1 μ. 0.1 μ is very compulsory for the nerve cells to remain alive. For sometimes it increases the level to transfer the nerve impulse from one cell to another cell. But if it is more than that, then the cell will go die. These two conditions very important here. And after the equation 8, we find this equation 11 to generalize here, to put all the values. Here I have used the finite element models. We discretize all these rectangular regions into the triangle using the triangle element, just to divide the regions. We find the variational form of the steady state of the equation 10. In equation 10, we use the shape function, equation 13, C1, C2, C3, that's a two-dimensional shape triangular element we have used. All these things we go throughout. Here the equation 20 we divide into IK, IM, just separate the terms, every terms here, and after that finally for the optimization of the equation, it will externalize our problem. We get di by du dot is equals to 0, and finally we get all the system of algebraic equation, because we are talking about the steady state equations only. Equation 38 we get the steady state system of linear algebraic equations, and we solve this system of algebraic equations. These parameters, particularly these parameters I have used here. If you go for the other vary from cell to cell, then we have to differ from the parameters. Here we find that voltage-gated calcium enter into the cell due to the voltage-gated channels. If in the presence of voltage-gated channels, it increases the calcium concentration. Here we see that it is 10 micromole, goes up to higher than the 10 micromole. In that condition, the impulse goes from one cell to another cell. In the absence of voltage-gated channel, it is quite low, up to 4 point something. In that case, nerve impulse could not move from one cell to another cell. This is a significant effect of the voltage-gated calcium channels. We see here. Then we use in the sense of voltage-gated channel, what is the effect of buffers. We take the different kind of buffers, different values, amount of the buffers, and with different association rate. There are so many different capacity of the different buffers. That's why we have used here. And after that, we see that at the less amount of the association rate, definitely calcium increase at the mouth of the channels, and again decrease and goes up to the that steady state condition 0.1 μ. In every case, we have to go to sell the Li. Here we see that the different amount of the buffer in the presence and absence of the voltage-gated channels. It means at different level we have to study what is the effect of the voltage-gated channels for the different kind of buffers. There are so many kind of buffers exist in our cell. In that case, in every case, we studied here that effect is significant in every case, but in the lower case, it is what? When the association rate goes high and high, or buffer concentration goes high and high, in that case, effect is not significant. It means buffer also plays very important role with the voltage gated calcium channel. This is the three-dimensional figure in the presence of voltage gated channel. In the sense of voltage gated channels, definitely we see it goes high and high. Here I have taken more amount of the buffers. That's why this effect is not seen significant. Here we studied for the different value of association rate by which buffers react with the calcium concentrations. What we see here, for the particular point of the at x is equals to 0, that is the mouth of the channel, x is equals to 1 micromole far from the source, 2 micromole far from the source, in the presence and absence of the voltage gated calcium channels. At the mouth of the channel, we see definitely it should be increased high and high. As we goes far from the source, it decrease and decrease. In the second and third, in red and green lines, we see it decrease and decrease due to the different kind of association rate. It means all the process occurs on the boundary of the plasma membrane. This shows the experiment also. All the process occurs on the plasma membrane. As it goes far from the mouth of the channels, and the process goes almost not effective. In fact, nerve vessels also exist on the area of the plasma membrane. That's why it is more effective here. It is conclusion. Only few figures I have shown here. Conclusion is here. We have studied the effect of voltage gated channels, effect of buffers, and comparatively we studied here. What we get in the presence of buffer, if the amount of buffer is less than the voltage gated channels, definitely plays very important role. As we increase the calcium concentration, or we increase the association rate constant, definitely the voltage gated channels not significant, but in the less amount of buffers, less association rate constant, voltage gated calcium channels definitely plays very important role. Due to this, our nerve impulse goes from one cell to another cells. Buffering is for the allied cells. That's why. This is the conclusion. We have used finite element because we have to discretize all the things. If you go for the different kind of source on the boundaries inside the cells, just like endoplasmic reticulum, if you want to go for this, in that case, definitely finite element is very useful. I think I don't know about analytic solutions for the different kind of regions is available or not. That's why I have used the finite element method, and that is the results. These are the differences.
Any question? Your equation is linear.
Yes, sir.
It can be solved analytically.
Yes, sir. Definitely it can be solved analytically. But when we go for the different source, just like internal source or the boundary different source, I don't think analytic solution